Kapag naririnig nyo ba ang salitang therapy, anong unang pumapasok sa isip nyo? Siguro masahe, o kaya naman ay pakikipag-usap sa isang counselor para gumaan ang pakiramdam. Pero paano kung sabihin ko sa inyo na may isang uri ng therapy na mas malalim pa rito? Isang therapy na umaabot hanggang sa mismong instruction manual ng ating pagkatao. And I'm talking about gene therapy. Before we dive into the science, I want you to imagine your body is like a high-tech computer. Most of the time, the software runs perfectly, but sometimes, there's a glitch in the code. A tiny error that caused the whole system to malfunction. In the past, we could only treat the symptoms of that glitch. Today, we're learning how to rewrite the code itself. So, what exactly is it? Based on our lesson, gene therapy is a technique that utilizes gene to treat or prevent diseases. Instead of using drugs or surgery, doctors can actually insert a healthy gene into a patient's cell to replace a broken or mutated one. Think of it as a delivery service. We use vectors, which are like molecular delivery trucks, to carry healthy genes into the body to do the job the abnormal genes can't do. To help you prepare for the rest of our report, keep these 3 strategies in mind. This is how the magic happens. First, the replacement. We swap out a bad gene with a healthy copy. Secondly, the knockout. We simply turn off or inactivate a mutated gene that isn't working right. Dito naman, kung may gene na pasaway o nagdudulot ng gulo sa katawan, parang pinapatay lang natin ang switch para hindi na siya makapinsala. And lastly, the third one, the reinforcement. We introduce a brand new gene to help the body fight off a specific disease. Dito, nagpapasok naman tayo ng bagong gene na magsisilbing sundalo para tulungan ang ating katawan na labanan ang mga sakit na hindi natin kayang talunin noon. And para mas malinaw, ang gene therapy ay parang software update para sa ating cells. Kung may mali sa blueprint ng ating katawan, nagdudulot ng sakit, hindi lang natin ito tinatakpan. Inaayos natin ito mula sa pinakaugat. But inserting genes is not always easy. To make this therapy truly last a lifetime, we need a special kind of blank canvas cell that can grow and stay in the body forever. That brings us to the ultimate partner in gene therapy, the stem cells. To explain how these incredible cells work and why they are the best choice for this field, I'm handing it over to my teammates. And good luck!
Imagine a world where we just don't treat the symptoms of a disease, but we rewrite the genetic code that causes it. This isn't science fiction, it's the reality of gene therapy.
Hello, I'm Kara Kennedy Taguino. Today, we're exploring how scientists use genes as medicine to fix ailments at their very source.
At its core, gene therapy involves 3 main strategies: replacing a faulty gene with a healthy one, inactivating or knocking out a mutated gene that isn't working right, or adding a brand new gene to help the body fight back. But for these genes to last a lifetime, we need a permanent home. That's where stem cells come in. They are the body's master cells with 2 superpowers: self-renewal, meaning they make endless copies of themselves, and differentiation, the ability to transform into specialized cells like nerve, blood, or muscle cells.
The real magic happens when we combine them in the lab. Doctors can take a patient's own stem cells, use a vector, often a harmless modified virus, to deliver a healthy gene and then put those cells back, because these cells live long-term in your bone marrow. They become a living factory that produces healthy cells for years. Here's a fun fact. Your body is actually a master of self-repair. Every day, your stem cells are working behind the scenes to replace your skin and blood cells as they wear out. In fact, the first successful gene therapy happened way back in 1990 to treat a young girl with a severe immune disorder, and it's been changing lives ever since. By merging genetic precision with the regenerative power of stem cells, we are entering an era of biological restoration. Again, I'm Kara Kennedy Taguino.
Today, I will briefly explain some stem cell jargons. First is potency. Potency refers to the ability of cells to develop the different types of cells in the body. The first type is totipotent cell. These cells can become all type of cells, including the placenta. Examples are the zygote, which is the first cell formed after fertilization. Second is pluripotent cells. These cells can be developed into almost any cell in the body, such as nerve cells, muscle cells, and skin cells. Third is multipotent cells. These can develop into limited range related cells. Example, hematopoietic stem cells in the bone marrow that produce different kinds of blood cells. And the last is unipotent cells can produce only one type of cells. Example that is spermatogeneal stem cells that produce sperm cell.
Stem cells. Stem cells are classified as follows. Embryonic stem cell or ES. ESCs are obtained from blastocyte, which is a very early stage of embryo. It is a ball about 50 to 100 cells and it is not yet embedded into the womb. The blastocyte consists of the inner cell mass, an outer layer of cells, and a fluid-filled space. The inner cell mass is a group of cells where the ES is delivered. Embryonic stem cells are promising because they are pluripotent and thus make all different types of cells in the body. Some are other facts about ES. ES are extracted frozen embryos left. ES are extracted from frozen embryos left over in vitro fertilization. Can be stored 10 years or more. Grow out ESC lines. Each line has a unique potency. Some problems are also taken into consideration in using embryonic stem cells. For ethical concerns, it is how ESC is being obtained and harvested to get an ESC line since it is a part of a human embryo. Another issue is to have a non-autologous match whereby it may not match the immune system of the recipient, thus resulting to graft rejection. It also have the potential to generate all the cell types in body, thus in the theory, there are no restrictions on the organs that could be regenerated. ES can also renew endlessly, so that a single cell line with carefully characterized traits potentially could generate almost limitless numbers of cells. Some of the identified disadvantages are as follows. Difficult to culture and grow slowly. Cells tend to develop abnormal karyotypes and other abnormalities. ES cells have the potential to form teratomas if all cells are not committed to the desired cell types before transplantation. Ethically controversial.
Adult stem cells. ASCs or tissue stem cells can often make several kinds of specialized cells. Also, tissue stem cells are found in different tissues of the body, such as bone marrow, skin, brain, and intestine. They help maintain and repair the tissues where they are located. Unlike embryonic stem cells, adult stem cells are multipotent. Multipotent, meaning they can develop into several related types of specialized cells, but not into all cell types in the body. Their main role is normal tissue, renewal, and healing after injury. So here are the examples or applications. Bone marrow stem cells produce red blood cells, white blood cells, and platelets. Widely used in bone marrow transplants for leukemia and other blood diseases. Skin stem cells continuously replace all the skin cells and help heal wounds and burns. Intestinal stem cells regularly renew the lining of gut, which wears out quickly. Neural stem cells, labeled as limited, can form some types of brain cells and are being researched for treating brain injuries and neurodegenerative diseases.
Oh, hell no, man. What the fuck, man? Get your ass on, man.
Hi guys, my name is Dela Prismat Andre R from BSE 2E, and our topic for today is induced pluripotent stem cells or iPSC.
What is induced pluripotent stem cells? Induced pluripotent stem cells are specialized adult cells that have been genetically reprogrammed to behave like embryonic stem cells. Who discovered induced pluripotent stem cells? In 2006, Japanese scientist Shinya Yamanaka discovered that introducing 4 specific genes, known as Yamanaka factors, into adult cells could reprogram them into pluripotent stem cells. How do induced pluripotent stem cells are made? Stem cells normally become more specialized over time through differentiation. Induced pluripotent stem cells or iPSC reverse this process by introducing specific genes that reset a mature specialized cell back to a pluripotent state. These reprogrammed cells can self-renew indefinitely and be direct to develop into many different cell types, making them valuable for research and potential therapies. Why are these cells important? The body's stem cells have the capacity to differentiate into a wide variety of cell types. They help the body heal itself. What is the relevance of induced pluripotent stem cells in STS, science, technology, and society? Induced pluripotent stem cells or iPSCs are created by reprogramming mature cells such as skin or blood cell back into pluripotent state similar to embryonic stem cells. This allows them to develop into nearly any cell type while avoiding the ethical concerns associated with the use of human embryos.
Umbilical cord stem cells. Stem cells that can be obtained postnatally from the umbilical cord. And also, umbilical cord stem cells are stem cells collected after birth from the umbilical cord blood, placenta, and amniotic tissues. These cells are multipotent, meaning they can develop into several different cell types such as blood cells, immune cells, and some tissue cells. Since the umbilical cord is usually discarded after delivery, collecting stem cells from it is non-invasive and ethically acceptable. The collected cells can be frozen and stored in cord blood banks for many years and for future medical use. So here are the examples or applications. Treatment of blood disorders. Umbilical cord blood stem cells are used to treat diseases like leukemia, lymphoma, and aplastic anemia by replacing damaged bone marrow. Genetic diseases. Children with conditions such as sickle cell anemia or thalassemia can receive cord blood stem cell transplants. Regenerative medicine. Cord-derived stem cells are being studied for repairing tissues in conditions like spinal cord injury and cerebral palsy.
Hello, I am Carljan L. Gumangan and I would like to talk to you about IVF surrogacy. Now, from what I know about IVF surrogacy is IVF surrogacy or gestational surrogacy involves creating an embryo in a laboratory using in vitro fertilization, commonly known as IVF, which in terms, you take the sperm and eggs from intended parents and then transfer them to a surrogate. Now, to get a better view of this or a clearer picture, think of it like this. You have an intended mother, which is the source of your egg cells, an intended father, which is the source of your sperm cells. You take those two samples, you put them together in the lab, and then you transfer that embryo to the surrogate or the person who wants to have the baby. That is the best example I can give. And based on my research, there are a few famous celebrities that I know that underwent through this, such as Kim Kardashian, Chrissy Teigen, and Sarah Jessica, which have all been known to undergo IVF surrogacy or gestational surrogacy. That is all. Thank you.